Hey, so today we're going to be talking about my enzyme experiment. So, for the introduction, I just want to talk a little bit about what enzymes are and what trypsin is. So, in terms of enzymes, enzymes are biological molecules that speed up the rate of chemical reactions, and they do this within the cells. So, the way that enzymes work is they break down the larger molecules to be more easily absorbed by the body, or they bind molecules together and make a new molecule. So why do we need enzymes? They are needed for metabolic pathways in the body. They're needed for respiration, digestion, and a lot of other life processes. And then when enzymes function properly, we can maintain homeostasis. So for our experiment, we use the enzyme trypsin. So just want to talk a little bit about what trypsin is. So trypsin is an enzyme that helps us digest proteins, and it's commonly found in the small intestine of humans. So when trypsin is catalyzed, it breaks down into smaller peptide bonds, So for the experiment, we were testing the question of at what salt concentration will trypsin show the greatest amount of activity. Our hypothesis was that there will be an optimum salt concentration at 1.2, which will show the highest amount of enzyme activity. And our rationale was that there are external factors that affect molecular activity. So as the salt concentration increases, the cells will start moving around and there will be an increase in activity. But we also knew that there was a salt concentration in which the enzyme would denature, so there would be an optimum, and then the optimum would drop off. So I just want to talk about the materials and methods. So why did we add TCA into our reaction? We added TCA to stop the reaction of the enzymes so we can measure the amount of substrate in the centrifuge tube. So why did we centrifuge the reaction after adding the TCA? And what was the pellet in the supernatant? So we centrifuged the reaction after adding the TCA because once the TCA is added, it allows the undigested substrate to precipitate, which forms clumps. So the centrifuging allowed for the collection of the pellets at the bottom of the microcentrifuge tube, and the supernatant was at the top, which is what we micropipetted out to test under the spectrophotometer. So what substrate did we use, and how did the addition of the sulfanilamide correlate to the measurement of the absorbance of 440 nanometers? So the substrate that we used was azocasein, which is a milk protein. The substrate contains sulfanilamide groups that, when digested, produce a red-orange color. And we used the spectrophotometer at 440 nanometers from the supernatant because of the added sulfanilamide groups. So we determined that high absorbance indicates high enzyme activity, and it's the opposite, so low absorbance indicates low enzyme activity. So why did we choose the control variables? We decided for the pH to be at 8 because that mimics the small intestine, and we decided for the temperature to be at 43 degrees Celsius because when we were looking at what other groups got in their results, we saw that 43 degrees had a high absorbance rate. So in order to test our complete optimum, we decided to try and test another optimum temperature to make sure that we were getting our optimum results. So how do we prepare the negative controls? We decided to set our negative controls at 0 degrees and keep the enzyme in, but we did see that that didn't really work. So what we tried to do was keep the enzyme in the ice at all times, keep the microcentrifuge tubes in the ice at all times, but unfortunately we weren't able to completely denature the enzyme. So our negative control values were still relatively high compared to other groups. But we did one trial of negative controls at each salt concentration, and we did 3 trials of each salt concentration. So we decided to use the salt concentrations of 0.3 molar, 0.6 molar, 1.2 molar, and 2.4 molar. And as I said, we did 3 trials of each and 1 trial of negative control at each salt concentration. So for our results, in this table you can see that 0.3 had the highest absorbance with an average of 0.704. And then 0.6 molar had an average of 0.627, 1.2 molar had an average of 0.511, and 2.4 molar had an average of 0.456. So we also tested our standard deviations, which they're all very low. So with 0.3 molar, it's 0.04, then it goes to 0.008, 0.06, and 0.044. So that proves that our data is relatively consistent because we have very low standard deviations. When standard deviations test how far away our data is from the mean, so we can see that our data is relatively close to the average, which means our data can be reliable. So for our t-test, we did not test against 0.3 because 0.3 was the highest, so we did t-test versus 0.3 molar, in which we got 0.032, 0.01, and 0.002. So that shows us that our results are statistically significant because all of our tests were below 5%. And our negative controls, as I said earlier, were a little bit high. We got 0.099, 0.067, 0.067, and 0.051. So they're very high compared to the other groups. And then our negative control values versus our t-test, they're very, very low. They're all negative times 10 to the negative 6 times 10 to the negative 7. So we got very low t-test, which shows that our negative controls are statistically significant. And then if you take a look at this graph, you can see that as we go along, our absorbance values decreases. So at 0.3, we have the highest, 0.6, 0.2, and 2.4, it goes down. And then you can also see at 0.6, the data is very consistent. We have a very low standard deviation of 0.008, so it shows that our data is very consistent. So in terms of our results, our highest absorbance values were at 0.3, where we had an average of 0.704. Our lowest absorbance values were at 2.4, where we had an average of 0.456. And we had the least variability at 0.6 with a standard deviation of 0.008. And all of our p-values were under 0.5, which shows significance. And then for us, in terms of our decreases, from 0.3 to 0.6, we had an 11% decrease. From 0.6 to 1.2, we had a 19% decrease. And from 1.2 to 2.4, we had an 11% decrease. So for our discussion, I just want to talk about the hypothesis and the experiment and our adequacy and any changes we would make. So we determined that the design of our experiment was adequate to test our hypothesis because we were able to get results that told us what the optimum salt concentration was. And even though that salt concentration did not line up with our hypothesis, we were still able to figure out which salt concentration was the best. So as I said, our results did not support our hypothesis because we hypothesized that our optimum would be at 1.2, but we actually found that the optimum was at 0.3. So as you can see from our numbers, the average absorbance values at 0.3 was 0.704 nanometers, while our average absorbance at 1.2 was 0.511 nanometers. So it's a very big jump between those two. So it was evident that our hypothesis was not correct. So I also said earlier that our results were statistically significant because all of our t-test values were below 0.05, which proves significance. So in terms of our temperature, we believed that 43 degrees Celsius was the optimum temperature to test our salt concentration at because we found that other groups said that 43 degrees was their optimum temperature. So we decided to test at 43 to try and optimize our enzyme activity. So in terms of variability, we found some variability in our data because not all of our data was cohesive. So for 1.2, we got values of 0.567 nanometers, 0.448 nanometers, and 0.517 nanometers. And although these values are similar, they're not completely cohesive. So it shows variability, but we also got a low standard deviation of 0.06. So it still shows that our results are significant, but they're not completely cohesive. And some improvements we made from our first experiment was we tried to keep the negative controls in the ice at all times in order to minimize our time of being in room temperature. So we saw that didn't really work because our negative controls were a lot higher as compared to the groups that did not add the enzyme. So next time we decided that if we were to run the trial again, we would just not add the enzyme because the negative controls were a little bit too high for what we were trying to prove. And we were also, another improvement we made was we were more precise when obtaining our supernatant. We made sure not to grab any of the pellet in our micropipette, and that was efficient to make sure that we were testing the right value in the spectrophotometer. And that is all I have.